Welcome. I'm Dr. Sunil Udani, and I will serve as your narrator through Chapter 6, Renal Physiology. In the first section, we will introduce renal physiology and go into the anatomy and the blood supply of the kidney. The kidneys function in several capacities. As excretory organs, the kidneys ensure that those substances in excess or that are harmful are excreted in urine in appropriate amounts. As regulatory organs, the kidneys maintain a constant volume and composition of the body fluids by varying the excretion of solutes in water. Finally, as endocrine organs, the kidneys synthesize and secrete three primary hormones: renin, erythropoietin, and 1,25-dihydroxycholecalciferol, or the activated form of vitamin D. Renal Physiology Section 1, Anatomy and Blood Supply. The kidneys are bean-shaped organs that lie in the retroperitoneal cavity of the body. In sagittal section, the kidneys have three main regions, as seen in Figure 6-1 on page 236 of the Costanzo text. The first, the cortex, is the outer region, located just under the kidney capsule. This is where the predominant number of glomeruli are located. The second, the medulla, is a central region, divided into an outer medulla and an inner medulla. The outer medulla has an outer stripe and an inner stripe. And finally, third, the papilla. The papilla is the innermost tip of the inner medulla and empties into pouches called minor and major calyces, which are extensions of the ureter. The urine from each kidney drains into a ureter and is transported to the bladder for storage and subsequent elimination. Next, I refer you to Figure 6-2 on page 237 of the Costanzo text. This figure outlines the functional unit of the kidney, the nephron. Each kidney contains approximately 1 million nephrons. A nephron consists of the glomerulus and its associated renal tubular structure. The glomerulus is a glomerular capillary network, which emerges from the afferent arteriole. The glomerular capillaries are surrounded by Bowman's capsule, also known as Bowman's space. This is continuous with the first portion of the nephron. Blood is ultrafiltered across the glomerular capillaries into Bowman's space. This is the first step in urine formation. The remainder of the nephron is a tubular structure lined with epithelial cells, which serve the functions of reabsorption and secretion. The nephron or renal tubule comprises the following segments, beginning with Bowman's space: the proximal convoluted tubule, the proximal straight tubule, the loop of Henle, which contains a thin descending limb, a thin ascending limb, and a thick ascending limb. The distal convoluted tubule is next, and finally the collecting ducts. This is outlined in Figure 6-2 as well as Figure 6-3 on page 238. Each of these segments has unique functions, and the cellular structures are particular to those unique functions. Each segment of the nephron is functionally distinct, and the epithelial cells lining each segment have a different ultrastructure. For example, the cells of the proximal convoluted tubule are unique in having an extensive development of microvilli, called the brush border, on their luminal side. Also, the space in between the cells, or the intercellular spaces, vary according to the segment of the nephron. This plays a role in the reabsorption of solutes in water, and whether reabsorption occurs through a paracellular route or strictly through a transcellular route. The brush border provides a large surface area for the major reabsorptive function of the proximal convoluted tubule. Other correlations between cell ultrastructure and function will be emphasized throughout the chapter. There are two types of nephrons: superficial cortical nephrons and juxtamedullary nephrons. These are distinguished by the location of their glomeruli. The superficial cortical nephrons have their glomeruli in the outer cortex. These nephrons have relatively short loops of Henle, which descend only into the outer medulla. The juxtamedullary nephrons have their glomeruli near the corticomedullary border. The glomeruli of the juxtamedullary nephrons are larger than those of the superficial cortical nephrons, and accordingly have higher glomerular filtration rates. The juxtamedullary nephrons are characterized by long loops of Henle that descend deep into the inner medulla and papilla and are essential for the concentration of urine. Next, we will focus on the renal vasculature. Blood enters each kidney via renal artery. The renal artery then branches into the interlobar arteries, then the arcuate arteries, and finally the cortical-radial arteries. The smallest arteries subdivide into the first set of arterioles, the afferent arterioles. The afferent arterioles deliver blood to the first capillary network, the glomerular capillaries, across which ultrafiltration occurs. Blood leaves the glomerular capillaries via a second set of arterioles, the efferent arterioles, which deliver blood to a second capillary network, the peritubular capillaries. The difference in arteriolar tone will ultimately determine the glomerular capillary hydrostatic pressure, the primary determinant of filtration occurring at the level of the glomerulus. The peritubular capillaries surround the nephrons. Solutes in water reabsorb into the peritubular capillaries, and a few solutes are actually secreted from the peritubular capillaries into the tubular lumen. Some medications are also secreted from the peritubular capillaries into the tubular lumen, specifically diuretics, which work on the tubular epithelial cells. Blood from the peritubular capillaries flows into small veins and then into the renal vein. The blood supply of the superficial cortical nephrons differs from that of the juxtamedullary nephrons. In the superficial nephrons, peritubular capillaries branch off the efferent arterioles and deliver nutrients to the epithelial cells. These capillaries also serve as the blood supply for reabsorption and secretion. In the juxtamedullary nephrons, the peritubular capillaries have a specialization called the vasa recta. These are long, hairpin-shaped blood vessels that follow the same course as the loops of Henle. The large surface area of the vasa recta serve as osmotic exchangers for the production of a concentrated urine. The flow of blood in the vasa recta also determines how well the kidney can concentrate urine. This concludes section 1.